Good afternoon to one and all. Is it visible now?
Yes, sir.
Yes, sir. Yes, sir. It's absolutely visible, sir, and audible also.
Okay. As you have mentioned, actually, I'm going to have a very tough task today because it's a mission impossible. The reason is like after a very nice lunch, people just tend to sleep and maybe they will start yawning. So just to keep the people who are awake, my talk has to be, a little bit interesting to them. So hope things are little bit, I'll try to keep it little bit interesting. Basically, I'll talk about very few basic things what we have been doing at JNU and other places. So our research lab mainly focuses on various 5 different areas I had given here: synthetic chemistry, bioconjugate chemistry, drug development, nanochemistry, and green chemistry. For my PhD students, I mainly focus on drug development and nanochemistry, and for the master students, because we have a MSc Biotech program, so in that the students will be mainly working on green chemistry. The bioconjugate chemistry is mainly I collaborate with other research institutions, and we do many of the bioconjugate chemistry through collaborations. Basically, Shankara Nethralaya is one place where we have already collaborated earlier, and this work, what I'm going to discuss today, is mainly focused on the work we carried out in collaboration with Shankara Nethralaya. And I'm also collaborating with NCL Pune and other institutes from USA also. Other than that, in the academic area also, I have been doing lots of teacher-centric and student-centric approaches. Basically, I'm a regular resource person for UGC MHRD programs at DIPSER, Sharda University, and KV schools. And I'm also UGC's subject matter expert and for various sessions, especially faculty development programs, so-called improvement program, in-service training are some of the things I do for teachers. And I also conduct workshops for the teachers. And in the case of students, I conduct basically open book examination because students' critical thinking can be improved by our examinations or the evaluations. So rather than rote learning, we mainly want to focus on students' concept-based understanding. So in our JNU, we are regularly conducting open book examination for the students so that they move away from the rote-based learning to concept-based learning. And I also use case studies, and I have developed a couple of massive open online courses which are currently available in Science platforms. So these are all the student-centric approaches I have been doing. So this is a brief introduction about me and our lab. In our today's presentation, we'll be talking about aptamers. So basically, what are aptamers? These are oligonucleotides, and they can be both RNA and single-stranded DNA or even peptide molecules. These are called aptamers. The major question people ask is like, what are they different from regular nucleic acids or other things? So there comes the meaning for the word aptamers. Basically, these are derived from the Latin name, which is aptus means to fit and meros means region. So these compounds are basically 30 to 40 nucleotide long sequences which fit very tightly. So that is what is the major name given for aptamers. And that's what here it is written. They bind to their target with the high affinity and specificity. See, this is one of the reasons. We will just look at other types of oligonucleotides, and why aptamers are given this name is simply because of their high affinity and specificity for binding. Their targets are generally proteins, peptides, carbohydrates, small molecules, toxins, and even live cells they can target. And they are actually, the binding is determined by their tertiary structure. So different type of oligonucleotides which are used as targeting biomolecules are they themselves for biomolecule include DNA, RNA, or non-coding RNA. Peptide nucleic acids are another type of oligonucleotides which are 15 to 18 oligonucleotide long sequences. These are used in antisense therapies. Locked nucleic acid also belongs to peptide nucleic acids. A similar thing here. The structure is little bit rigid. That is why this is called the locked nucleic acid. So these are basically small sequences, like less than 30 nucleotide sequences, And aptamers are little bit bigger, so they are like 40 to 60 nucleotide sequences are generally used in the case of aptamers. Due to the longer sequences, these are much more specific binding and they can easily recognize the targets much efficiently. So they are little better than peptide nucleic acids and long-chain nucleic acid. And in the aptamers, we have xenonucleic acids. And of course, people know about small interfering RNA. So these are various different types of oligonucleotides which are used for drug delivery developments and targeting therapeutics and various other areas. So what are the main applications of aptamers? They can be used as a new drug targets or for bioimaging. So in this presentation, I'll be mainly using or talking about the bioimaging aspects of aptamers. They are also used as a therapeutic tools, a drug delivery, diagnosis, and food inspection. So aptamers are finding applications in various areas. Today, basically, I'll start with some fundamental aspects of what is aptamer synthesis. Basically, the aptamer synthesis, as I mentioned earlier, there is a sequence like 30 to 40 or maybe up to 50 nucleotide long sequences are identified, and they generally undergo three-dimensional structure. That's what we have seen in the first slide, like specific targeting is achieved using the three-dimensional structure. So the sequence is basically converted into the three-dimensional structure. So the primary structure is converted into the tertiary structure. Some softwares are generally used for doing this. This is basically the bioinformatics work people generally do. So the whole process of the synthesis basically starts with the bioinformatics work. And the bioinformatics work basically identify the three-dimensional structure of the particular aptamer. And whether this particular aptamer can fit into the target is the main thing we are actually looking for. So the receptors which are present in the cell are our final targets. So this aptamer, if they can fit very nicely into this particular receptor, that is what we are actually looking for. This is a basic initial step of the synthesis part. It starts with the bioinformatics work. And once we are done with this identifying the target binding site, then our main aim is whether the sequence is the right sequence, then we have to synthesize the sequence. So there are commercial products are available where they can actually get it, like Alrich and others are available, so we can send them or ask them to prepare the sequence. So they prepare the sequence and give it to us. So this is how we start our process. And an ideal aptamer has to have a high binding affinity to its target, and the specificity is also very important, and stability and bioavailability. So these are all the four basic fundamental things which are the desirable properties of an aptamer, which for any biomolecule, if you are going to target cell-based approaches, then these are all the basic requirements which are essential. And in the case of aptamers, one special technique called SELEX technique is the one people generally follow. So this is basically the systematic evaluation of ligands by the exponential enrichment. Basically, when we talk about this particular sequence, we actually have only one single sequence. So with this one single sequence, even if we make that aptamer, can it be very specific for the target binding, and can be used actually for our imaging and other studies. So now we do need a large quantities or large amount of the substance. So how do we make this particular sequence? Because we cannot make a large amount by purchase from outside. So that is where this particular process comes into picture. So basically the selection of the ligand sequences that binds to the target is identified in the first step. And in the second step, this particular sequence is mixed with non-sequences, and by affinity methods, we can easily separate the aptamers from the non-aptamers. And then these identified or the isolated aptamers were amplified. So this is start with 1, then go to 10, again repeat the process start from 10, go to 1000, then again repeat the process from 1000, you make multiple copies of this aptamer. So this is what is the major thing that is done. So this is basically run multiple times. When we do this multiple times, basically that is what is the systematic evaluation of ligands by exponential enrichment. So this is the crucial part for the aptamer synthesis. And here I will just show a brief sketch of how the process is actually done. So when we are going to find out the aptamers, many times what happens is the sequence may have some changes, or maybe few modifications may also be present. So in the initial library, we are actually going to have multiple sequences. Even though the bioinformatics work does give some rough idea about the tertiary structure, but not necessarily the tertiary structure is the one actually going to be seen inside the in vitro selection process. So all the times the solvent polarity and the various other factors are very crucial for the binding. So we do need to have a very specific target for our further studies. So we need to identify, even though we have a library of sequences, we do need to get the exact sequence, and that is what we are going to multiply a number of times. So the step one is basically we need to have the target onto which this aptamer is going to bind. So once we have the target and add this library of the aptamers to this particular target, then if you see the green one and this blue one and maybe this light purple color one. So these three sequences actually bind to this target, but there are other sequences which are not binding to the target. So this way we are actually going to identify only those targets or those sequences which bind to the target is the only thing we are mainly focused on. So what we do is once the sequence binds to the target, our next effect is we have to remove the unwanted things. That means the non-binding sequences have to be removed. So this is the next step we do. So during the incubation process, the binding becomes a little bit stronger, and the unbound sequences are removed in the first step. So only the target with bound sequences. So this is the one we are starting off with our initial stage. So once we have this particular step, then the next one is we have to do the separation. That means the bound sequences has to be removed from the target so that we have the right sequences which are binding to the target. So separation can be done. In that process, the target is removed and the only we are getting the binding sequences are eluted. So I will show about how the separation can be done. Affinity chromatography and various other methods are available by which the target and the sequence can be separated. So this is the first pool of sequences which we have identified. So 1, 2, 3, these three sequences are maybe the fourth one which is on this side. So these four sequences actually bind to this particular target. So this is our initial pool. Then once we have the initial pool, what we have to do is we have to amplify this. So run the PCR to amplify all these things, and then we get multiple copies and repeat the same process several times. In that way, we can actually increase the specific binding of the particular sequence multifold, and we will actually get the right sequence which are highly binding to the specific target. So this is what the amplification, that is what the exponential enrichment, the last word in this SELEX is the one which is done in the sixth step. So this is the repeated steps multiple times we carry out. We can put it like first expose the sequence to the target, then allow it to bind, then wash the unbound sequences, then elute the bound sequences, amplify, repeat. So this way, systematically we are actually creating the particular ligand. Here nothing but the sequence by multiple exponential enrichment. So this is what is the whole process by which the aptamers are generated. Then the purification, as I told, there are various separation methods given here: nitrocellulose membrane filtration, and of course, being a biotechnology student, you already know about that. So I'm not going to talk more about any of these things. So some of the methods which are given red are somewhat difficult to carry out. And here I will just talk about some of the separation methods, nitrocellulose-based membrane filtration techniques, or affinity chromatography, or magnetic bead-based separation principle. So the basic principles are given here. In the case of affinity chromatography, we separate it basically based on the biological interactions. That is, receptor ligand or antigen antibody interaction is used. That is what is the affinity. So by that we can actually separate this out. And the limitation with affinity chromatography is like if the target lacks the affinity tag or the functional group needed for coupling to the beads, then we cannot use it. Then the other method is the magnetic bead based one. So here the target is immobilized via some interaction or a chemical reaction basically, and then the separation is carried out. So here some pictorial representation of the same is given. So some of the functional groups which can be used for the activating the beads are given here. There is an alcohol unit, epoxide, amines, streptavidin or aldehyde carboxylic acids, and various other groups are given. So these can be used for binding the targets. And here the magnetic bead based one, like here we can use magnetic separation for these kind of aptamers which are bound to that. And we can also use the affinity column for the separation. So these are all the various separation methods by which we can actually separate the library, whatever is attached to the target. And this is like we are doing it basically in vitro. And if you want to go with the cell SELEX case, in which we can also use a whole or the live cells can be used in the cell SELEX method. In the case of normal SELEX, it doesn't work on extracellular proteins. So this is one of the limitations. And this also doesn't work on live cells. And here, when we talk about the in vitro purification, this also does not work on full length proteins. So these are some of the limitations of SELEX, which can be overcome by the cell SELEX methods. So here, we don't even have to have the prior knowledge of the cell surface marker proteins, or we can also use it for the live cells. One important thing in the cell SELEX is basically the limitations with aptamers are given here because they generally repel each other, because the backbone, which is a polyanionic one, and the cell surface sometimes repel each other. And they also non-specifically interact if there is a positive amino acid that is present, like arginine or lysine. So these are some of the limitations of using aptamers. So we have to be a little bit careful when we are selecting our targets, because if we wrongly do the selection process, then we will end up with a compound which is binding but not specifically binding to the receptor. And some important method is called a negative selection step. So I'll give a small analogy about a negative selection, because the negative selection is one of the most crucial thing which is generally used by especially women. When a woman want to buy a new cloth, what do is generally they follow this particular negative selection process. So in this negative selection process, what they do is they go to the shop, and what they are looking for is not what they actually want to purchase. They don't want to purchase what their neighbors are having. So that is what they do in the purchasing of materials. If the neighbor is giving peel color dress, then they will make sure like they don't want to buy the same peel color. And if the neighbor is having some maroon color or maybe some magenta color, then the lady actually looks for avoiding those colors. So that is what is called the negative selection. That means they don't want to choose the right color in the beginning, but they want to avoid the negative things that means their neighbors are having. The same way in the cell SELEX, what we are doing is a negative selection. In this particular case, what we are doing is we are not identifying what is actually binding. What we are doing in the negative selection is what is actually not binding. That is what we are actually going to select. So in this particular cell, we have to use a non-target cell. So that means very similar to the neighbors are used. The same way, we are not using the right target. That is the dress what they want to purchase. They are not targeting in the beginning. What they are doing is the negative variation, where they are taking a non-target cells in the beginning, and the library is actually passed through the negative selection process. In this, the irrelevant pool is actually removed by the non-target selection, and only the unbound one which actually the person wants to purchase is selected. Then we do the positive selection in the second step. So when the unwanted colors are removed in the first stage, then we have to only select the right one. So this is how the negative selection starts, and some limitations of cell selections are given here because live cells are very important, and proper cell culture techniques are also essential. So with this, we will just come to the conclusion of what are all the basic process and how we can do the aptamer application part is given here. So they can be used for cancer imaging. Some of the examples are given here, and various aptamers are currently in the clinical trial. Chemical aptamer can also be used as a chemical antibody. Some of the important points are given here. And chemical modification of the aptamer actually gives additional important properties which are required. Say, for example, if the aptamers are not having a good renal clearance, then we can actually make some modification or add some PEG groups so that they are removed from the body very easily. And improving the specific binding, we can also make some modifications to the nucleobases, or we can make some modifications so that it is resistance to nucleus degradations. And these are some of the modifications we can carry out on the aptamer, because these are basically the nucleic acid nucleotides. So here we can make some modifications on the chemical structure table. So some examples are given here, some modifications like introduction of the hydroxy group, amino group, fluoro or methoxy gives more stability to the aptamers in the biological environment. So some examples are advantage of antibody over aptamer is given here. I'll quickly go through that. We will just move to the. Our work was actually published in the Chemical Communications inside cover. So this is the work strain promoted acid alkyne cyclization reaction is the one we are going to see. The major thing is we have been using bioorthogonal chemistry for carrying out all the conjugations and other things. Basically, the bioorthogonal chemistry started with the interface between chemistry and biology. So here, chemical modifications of the biomolecules are done. So chemical modification of biomolecules. So this is the biology part, this is the chemical part. So these two combined together, we have the chemical biology aspects. In traditionally, what we do is modification of protein is done with the side chain modification, especially cysteine, lysine, or tyrosine, or maybe tryptophan. The advantage for this is basically lysine can react with activated esters, sulfonyl chlorides, isocyanates, or aldehydes to give various derivatives. So these are used as a chemical modification of the biomolecules. Tyrosine tryptophan can also be used for the transition metal mediated process, native chemical ligation. So these are all the previous methods people have been using for making some changes to the biomolecules. In the case of but they do have some of the limitations, like the molecule, which the biomolecule has to have a single site for protein modification. Otherwise, if more than one site is there, all the site will be modified, which is not required for us. So, and again, the residue also need to be on the surface. If it is in the interior part, then the modification cannot be done. And the dimerization, solubility problems, and non-specific hydrophobic interactions are some of the limitations of the traditional methods. In the bioorthogonal reactions, what we do is the biomolecule is basically incubated with some metabolic precursor containing functional groups. So this is the most crucial part. So a chemical entity is added to the biomolecule in the first step, and in the second step, this is the probe is reacted with the chemical entity, whatever is that. So this is what is basically called as a bioorthogonal reaction. So we take biomolecule, add some chemical entity, and then attach the probe to this particular chemical entity so that all the three are put together. So one such example is reaction of acid alkyne reaction. So here alkynes, especially the cyclooctane, is reacted with an acid to form the triazole derivative as shown here. So an example is given here. This is basically a 1,3 dipolar cycloaddition reaction of acid. So this is the acid unit. This reacts with the alkyne unit in the presence of a copper catalyst to give this triazole derivative. So this is the most crucial reaction we have employed in this particular sequence. So can you see this now?
Yes, sir. Yes,
Yes,
sir.
sir.
Yes, yes, yes.
Okay. So we have done the strain promoted acid alkyne cycloaddition. So this is the acid part and this is the alkyne part. So this alkyne is basically this acid part is the one, the chemical entity we were talking about. So this is the biomolecule, whatever is the one here, basically the aptamer. So the aptamer is now added the acid unit, and this acid unit is reacted with the alkyne. So this alkyne has the fluorescent probe. So this way what happens is when the entire conjugation takes place, so the aptamer is going to bind to the target, and then our whatever is the fluorescent probe can be used for finding it out where the aptamer had actually bound to the biomolecule, basically that is the target. So bioorthogonal reaction can target biomolecule is already known. And EPCAM is basically epithelial cell adhesion molecule is overexpressed in various cancers, and EPCAM aptamer is also used for cancer targeting. So these two are isolated things, people have done it separately. And what we did are the missing link was like aptamer based bioorthogonal reactions targeting EPCAM positive cancers are not at all known. So we connected these two these two compounds using our link, and then finally we created our work here. So the sequence is basically known for EPCAM specific sequences. That is, this is used as a probe, and then we also have a specific functional group. In this particular case, we have added acid to the aptamers. So the aptamer sequence, what we have used is given here, SYL3C. And the functional group, what we have added is the acid functional group. And the probe is actually having the Alexa fluor is attached to the fluorescent dye is attached to the probe, that is DIBO, that is dibenzooctane. So I will show the structure here, the next one. So here the fluorescent tag is attached to the DIBO, and this part is a separate unit. So this entire unit is a separate unit, and this is the other unit. And these two units are basically connected via the conjugation. So this is what we have actually done that. So the aptamer has the acid unit which is present, and the DIBO is having the alkyne, and the DIBO is having the fluorescent probe. So when combining these two, we get the triazole moiety. So this is the linkage that occurs in exactly between the acid and the alkyne. So that is what we call like a spark reaction. And once we connect all these things, this target, this aptamer is going to attach or bind itself to the cancer specific targets, and this is the fluorescent probe. So once it is attached, we can actually see that whether it is attached or not through this particular probe. So this is our overall work what we have done here, and using a normal chemical synthesis, we have joined these two things, and this reaction is a very, very facile reaction, that is reacting an alkyne with an acid to form the triazole can be carried out at room temperature, because many chemical reactions, if they cannot or if they require a high temperature, they cannot be done inside the body, because biochemical reactions require room temperature reaction, and they also should work in the biological systems like a PBS buffer or very similar things. Then only we can actually use this for the biological studies. So this reaction we have specifically chosen because this is a very, very highly reactive reaction, and we can actually use a copper catalyst. I will tell the reason why we have to use a copper catalyst, because this reaction combination of DIBO and the EPCAM aptamer is chemically shown here. When these two are combined together, we get the stable triazole conjugate. So the formation of this reaction can be studied using HPLC techniques and MALDI and other methods to confirm the product formation. So how we run the reaction is we have did the 1:1 ratio or 1:2, 1:4, 1:6 or even higher ratios. We have combined these two things, just mix these two things at room temperature. That is the only thing the reaction actually works. So when we run the reaction at 1:1 and this is the DIBO one, when we add more amount of the DIBO, what happens is basically separation becomes very, very difficult. So the conjugate generally don't form that easily. So we have to avoid using large amount of one of the reactant, that is the DIBO has to be used in 1:1 ratio. So here we get the complex which is formed after the reaction. So this is how the imaging what we are going to see. See, this is the cancerous cell, and the cancerous cell has a particular marker or the receptor at the end, and this particular aptamer with the fluorescent tag is going to bind to this particular receptor, and then by that we know exactly where it is bound, and from that we can actually identify what are the cancer cells. So the step of the aptamer conjugate was done using HPLC. So this is at two different UV wavelength. The traces are given. One is at 260 nanometer, another one is at 594. So you can see the conjugate is appearing here. The DIBO appears very in the early elution time at 15 minutes, and the conjugate comes very late at 32 minutes. And here again, these two are nothing but different UV traces. And the purified compound was analyzed by the MALDI. So in the MALDI, aptamer alone has a 15,000 molecular weight, whereas the aptamer plus the conjugate has 16,321. So with this, we can actually conclude that our reaction had actually occurred. And once we have this, then the next one is we do need to know the stability. Whether under the physiological conditions, whether there is any breakage between the fluorophore and the DIBO, is there any breakage here? Or is there any breakage between the aptamer and this unit? So this is very, very essential. Otherwise, what will happen is if the molecule breaks within the body, then we have no idea of knowing whether the compound actually reaches the target, and what we are actually seeing is the right aptamer based targeting. We cannot do it. So we have to make sure before we actually go to the next step, we have to do the stability study. So that's what we did. So the compound in PBS was put at physiological temperature and pH for several hours. So up to 72 hours, we checked using aggressive gel electrophoresis. We found the intact compound. So that means the conjugate is stable. And dialysis also did not show any degradation or release of either DIBO with the fluorophore or just the fluorophore alone. So these two studies clearly indicated that our conjugate, whatever we have made, is quite stable. So this added some more additional proof for us to move to the next step. So there are two major type of interactions which are possible. We have chosen nucleolin aptamer, and the nucleolin is present both in the cell surface and inside in the nucleus also. So we do need to know whether the aptamer interaction, what is happening is either membrane bound or it is internalized. So these two things we have to study very carefully. So the membrane bound thing can be studied at incubation at 4 degrees, because at low temperature the aptamer cannot penetrate the cell wall, so it will be only found at the membrane only. So the internalization does not take place if you incubate at 27 degrees centigrade or 37 degrees centigrade. So by doing the incubation at two different temperatures, we could actually differentiate between the membrane bound and the internalized one. So for the membrane bound, we used flow cytometry, and for internalization, we used fluorescent microscopy. So these two are the two different methods by which we can actually see how the aptamer is bound to the target. So here is the cell surface binding studies of our conjugate. So the first one is the control, second one is the DIBO alone, third one is the conjugate. So here if you look at the values, we have a very good binding here in this particular cases. So the binding is much more compared to the either DIBO alone or in the control. we have used different cell lines. So the very RB1 is the retinoblastoma cell line, and MCF7 and PC3 are the other cell lines we have used. In all the cases, the conjugate had a much higher binding. So this is confirmed, and we also did the flow cytometric analysis. In this, you can see the pink color, the MCF conjugate is shown here, and this is only the MCF7 conjugate, but DIBO with our particular conjugate is shown here, and the unstained ones are shown here. So from these images, we could say like the binding to the cancer cell line is much more efficient, that we can confirm from these images also. So these two had actually shown that our conjugate, whatever we have synthesized, is actually working very effectively. And we also did the inverted fluorescent microscopy images of the conjugates. So PC3 cells with EPD DIBO, and these are all the compounds, that is the conjugates, whatever we have done. And in that case, you see the red color. The red color is basically from the fluorescent dye, whatever we have used. So this clearly tells that the membrane bound, if you look at in these some of the structures, the red color is only at the outer surface. So this clearly tells the binding actually takes place in the cell surface. And here again, if we look at inverted fluorescent microscopic images, so here you see the very faint color that is present here. So with this, we can actually conclude that the membrane bound nucleolin receptors actually interacting with our aptamers. And we can also confirm that using these kind of techniques, we can actually study the cancer cells, which are all the places where the cancer had spread, we can actually study. So here you can see the internalization of the same conjugate. So instead of the bound one that is at the surface, in these images you will see this is present actually inside the cell. So we can also study both the internalization as well as the membrane bound cases using our conjugates. So the conjugate, by incubating at different temperatures, gives different images. So based on that, we can actually confirm the internalization as well as the cell surface binding can be studied using this particular aptamer based conjugates. So here again, couple of other examples. And we also studied about the cytotoxicity, because whenever we do cancer based studies, the compound or the conjugate should not have cytotoxicity, otherwise our entire study is of no use. So we also did the cytotoxicity studies and found like, up to 1 millimolar concentration, 1 micromolar concentration, we don't have cell proliferation. That means cytotoxicity is not very high. We have confirmed it. So this, in other words, tells that like up to micromolar concentration, we can use our aptamer conjugates for our studies. And we also studied the serum stability studies. These are regular studies people generally carry out to see the stability of the bioconjugates in the biological environments. So there is a 70% degradation after 36 hours. So up to 24 hours, there is no significant degradation. So we can actually study for up to 1 day, we can easily study that. Afterwards, there is a degradation. And if we have to use the same aptamer for a longer duration of studies, then what we can do is we can do some modifications, like the chemical modification of the aptamers can be carried out with the 2' OMA group or LNA, or other substituents can be appended to the aptamer, which will increase their stability in the serum for longer time. So with that, I conclude my talk, and this work was carried out in collaboration with the Vision Research Foundation, Sankara Nethralaya. Thank you. I am open to the questions.
Yeah, thank you so much, sir, for such outstanding, excellent, informative talk. Even though it did not left any corner to ask any question, because you have touched all the aspects from initial point to extraordinary exploration. But still, if any question from the guests, delegates, then it is most welcome. So please ask if any kinds of
Yeah,
query.
yeah. Can I ask a question?
Yeah, sure.
Yeah, Professor Balaji. First of all, I just congratulate that this is a very thought-provoking lecture. I have listened, uh, and I heard a lot about the aptamers, but you have presented the aptamers chemistry in such a nice, simple manner that it caused interest in me to do some work in it. Actually, we work in CBR and defense, and we love to see some markers, like for example, for like have you heard about the toxic industrial chemicals? So so somehow if those chemical got internalized because of some accident or because of some mishaps, so it caused some damage biomarkers. So can we use aptamer chemistry to use to develop some biomarkers, some biosensors, so that we can actually quantitatively as well as qualitatively determine the level of damage markers inside the animal model? Can it be possible?
Yeah, it is always possible. The only thing is, like as we have mentioned in the beginning, see, these aptamers means basically they bind to the target very specifically and very strongly. So we do need to know, only thing is like the cell surface should have some receptors, and we need to
Okay.
identify the receptors. So that is the only thing is most important thing. And in our work, we actually know about the nucleolin aptam. I mean, basically the nucleolin is known. So that is a
Yeah.
protein we actually have been targeting. So since the protein is known, we exactly know the 3D structure. And then designing a particular marker, especially aptamer for this one, was taken later on. So the initial step for the entire process is you do need to have a good marker which you can actually identify on the cell surface. See, if it is internalized, it's very difficult and we cannot actually start our work. So the cell surface markers are the most obvious choice and the easy one for you to target. And then we can actually use, see, this is basically a random things you have to do. Like if you are a bioinformatics person, you actually know that, like, I mean, not all the work what we do is actually translated into the animal based model or human trials, because we have to
Yeah.
do so many other studies. So at least
Yeah.
the initial thing can be done using some of the softwares like Mfold, and there are various docking softwares that are available. So you can randomly look at the sequence, and maybe up to 30, 40 or 50 nucleotide long you can take, and you can also use BLAST and identify the proper sequence also, that is possible. Then you can actually go for, like, if you really want antisense technology is also known. So peptide nucleic acids and other things people generally use for antisense technology. And aptamers can also be used for the same purpose. So the receptor identification and binding to the receptor are the most crucial part. If you can successfully achieve that one, then any aptamer can be used. Then then once you know the sequence, it is actually like, I mean, a laborious work of repeating, and get more number of your aptamers and then do the studies. So the initial kit, that means binding, is the most crucial one.
Thank you so much, sir. Thank you so much.
Sir, Professor Sharma sir, Head Department of Biotechnology. Sir, any queries or any question? Some words about the speaker, sir.
Thank you, Dr. Balaji, for such an enticing talk. Yeah, thank you for making us aware about this aptamer technology. I think in current scenario, it holds lots of promise, and from your observations also, it seems a very, very promising technology. And I congratulate and thank you also for delivering such a wonderful talk. Thank you. Thank you so much.
Sir. Thank you so much. Thank
Sir,
you for giving me
now
an
I would
opportunity
like
also.
to invite
Thank you.
Professor Sharma sir and Professor Ojha sir. Please felicitate to Professor Balaji sir by life membership of International Association of Zoologists. So please, sir.
Many congratulations,
Many
Dr.
congratulations.
Balaji.
Thank you, sir. Thank you
It's
so
a proud
much.
honor for us.
Thank you. Thank you so much.
Thank you. Many congratulations.
Congratulations, Professor Balaji.
Thank you, sir. It's really a great honor and a surprise and a privilege for me to receive this life membership certificate. I am really thankful to you people. Thank you so much.
Thank you so much, sir,
Thank you.
for giving time for
Thank you.
us. And now we have a short break for 15 minutes once again. And thereafter we have an renowned personality, Professor A.K. Saini sir, is a renowned worker in cancer biology and acts as scientist in NIH also. So we will meet him after 15 minutes at 3:15, I suppose. And till for this time, we are at short break. So thank you, sir,
Thank you so much.
for giving
Thank
time.
you so much.
Thank you.
Thank you. Thank you.